All right, so we're recording. Okay, so to start with, just for our biology final, I know Caden's in the classroom, but for all you guys that are going to watch the recording, this will help you answer the study guide sheet. And also, make sure you guys know the final is 20% of your grade, and it has to be proctored. So please find your site coordinator or whoever you signed up with and find them and schedule your final. And our goal this week is we have 1 week before we can start taking our finals is to get our grade as high as possible. I still have some bonus points to add into your guys' grades for you guys that posted to the discussion boards. So I will add those in. And then if you complete the study guide, you can get bonus points. And then one other opportunity I have for you to get bonus points is I think this morning you guys should have got an email with course evaluations. I can't see who completes what course evaluation, but I can see who completes it. I won't see your responses. So if you complete that course evaluation, I will also give you 10 bonus points. So that's another way you guys can up the score. Did you get it in the email, Caden, this morning?
Yeah.
Okay. So, and then please encourage your parents to help take that. It gives us feedback. Answer it honestly. It helps me improve as a teacher. If there's difficulties you had in the course or something we can do with IDLA to fix the course, let us know. And I know summer is really fast paced. It's kind of how it has to be to get a 16-week course in over the summer because we don't have as much time during the school year. So that is always a stress for kids, I know, but just give us our feedback of what could be different to make you learn biology. So with that, we'll get into it. Our final review. I just have 14 slides, so it's not going to be really long, but hopefully helpful. I kind of made one summarizing each 1 to 2 slides for each unit. Our first one for sure that you guys have to know are the characteristics of life. So make sure you know what each of these are. I think, of course, everybody knows that living things have to be made of cells. Be familiar with that word homeostasis, which means to keep even. So like on days today, like when it's 100 degrees outside, our bodies are sweating and figuring ways to cool ourselves off so our body can maintain homeostasis and keep us cool. And we all have a metabolism or use energy. All living things have DNA. All living things, they don't have to reproduce to stay alive, but for their species to continue, all living things have to reproduce to continue the species. And then living things change. Evolution and adaptation is they change their environment. If the environment changes, living things have to change or they can't carry on. And then the last one is response to the stimuli or response to the environment. Just like how a sunflower rotates throughout the day, following the sun, or we blink when somebody gets something real close to our eye. All living things have some kind of response to their environment. So make sure you guys know those. I did put a slide in here and I sent this out to you with each one of those. You can print that off. It'll help you. Some common vocabulary that are within those characteristics is make sure you know the difference between unicellular and multicellular, which if we think of a unicycle, it's one wheel, so one-celled or multicellular. And we know the difference between asexual and sexual reproduction. Asexual means it's a clone, kind of, the exact copy, like strawberry plants and raspberries and a sponge that cuts off. It's exactly, we can cut a part of it off or it can grow without reproduction. Sexual reproduction is like what we're studying right now when we're studying genetics. We combine those genes. And so we get the offspring looks different than the parents. Under metabolism, make sure you know autotroph and heterotroph. I teach my students autotroph is like automatic. It automatically makes its own food. We are not autotrophic. It would be really nice if we could go sit out in the sun, absorb rays, and get food, but I would miss eating. I kind of enjoyed some ice cream today when it was really hot. So that makes us a heterotroph. Is this all making sense, Caden? Am I going too fast?
No, you're good.
Okay. So heterotroph, something funny I tell my face-to-face kids is you can go home and call your brother or sister. You can be like, you heterotroph. And they won't really they should know, but all it means is we eat. And so it's a big scientific word, but it's just to help you remember that vocabulary. It just means we eat other stuff to live. We cannot photosynthesize. And so I went over homeostasis, which means we keep even. And then we did a whole unit for the hereditary unit on DNA, which I'll get more into in a bit. But make sure you know it's the code that lets all organisms make protein to make them. Then under respond to stimuli, we can respond to living and non-living things. The two vocabulary words there are biotic and abiotic. In biology, a lot of times in science, when you see the word a, it means not. So abiotic means not living. And then I know one of our test questions, if you guys go through your old tests, one of them was to ask you, what's the difference between growth and development? And growth means we just get bigger in size, and we do that by getting more cells. Our cells don't just get bigger, we get more cells. And then, sorry. And then also, if we look at it, where was I at? Sorry, my phone rang, but I should have asked. Development. Development means we change. If you look at a baby has a really big fat head. Well, as adults, we don't still have really big fat heads compared to our bodies because we develop. And our adult form of our body looks a lot different than the baby form. So we change forms. A really common one is a caterpillar to a butterfly. That caterpillar, that's development. It totally changed its form. And then basically knowing for the last characteristic of life that all organisms have to adapt. They have to have something to let them go on to make sure that that species survives. So those are the characteristics of life. Be very familiar with these. I know there's test questions from Unit 1 on those that you could retake. And that's another way to up your grade. Caden, on this one too, is if you didn't score well on a test or somebody didn't score, go retake those tests. You can retake all the tests, and if you need it opened again, I can do that too. Okay? So make sure we do that. And hold on just a second. I think I have a student trying to get in that can't get in. Is it okay, Caden, if I help out here for a second?
Yeah, that's fine. That's fine.
I'm just going to send him a quick email to, it says it's not letting him in. Caden, did you just click on the link in the calendar? Is that how you got it?
Yeah, yeah. It was in the email, yeah.
Okay. I'll have him go that way. Maybe he's trying to get into the class. I'll just send him. If it'll let me. Hold on just a second, Caden. I'll help him in, and then we'll be.
All right.
You're still there, right?
Yeah, I'm here.
Okay. I'm going to try to reply to him on my phone so I don't mess up this recording. I might. Okay, so I think that'll help him get in. Who's that? Right here. Okay. Let's see if he can join us. I don't know. He's not in here yet, but he just emailed me. You just clicked on the link in the email?
Yeah.
Hayden, I'll tell him that. Maybe that. Let's see here. Is everything I'm going over right now still helping you?
Yeah. I'm just taking some notes here.
Yeah, and fill in the worksheet as we go a little bit. You can try. I don't know why it's not logging in. Okay. And I'll try to send him a new link. It's kind of tricky to pull up two. I got it here, but. I'm going to copy the link for him too. I know I just posted it to the announcement.
Oh yeah.
In the updates. But I think some of the things they're definitely to help you out. Have you make sure you take all the unit tests and then come back and you can retake them because most of the time a lot of the test questions for your final are going to be pulled from your unit test.
Yeah.
So that's a good way to get study and to help out. So I'm trying one last time. He's still struggling here. I'm sending him the link. Then we'll move on. He says, goodbye. I know. Okay, that one. Wonder why it would give him problems. You got it, Caden. Sorry, Caden. I'll keep moving here if we don't get it.
I'm good.
And sorry for you guys watching the recording. We can fast forward through part of this, but it should work. All right. So we'll move forward from here as we're going. I sent it twice, but if it works. And no, good. Okay. All right. So that's basically unit. A lot of unit 1 is basically characteristics of life, and that's what biology is, the study of life. And it sounds pretty basic. Like, why would I study this? Like, is it not very important? But just to give you some justification why you should study this stuff, this is what we're looking for when we're looking for life on Mars and like other planets. What does define life, and how do we find it? So even though it sounds really basic, like the life is living things, it's really more complex than that because we know what we think life is, but when we go look on other planets and everywhere else, it could be totally different. So this is what biology we have said life is, and so when we think of it that way, I think it's pretty cool. It's pretty cool to look at all the different things from one-celled organisms to complex things like an elephant or a giraffe or I mean, even protists and amoebas, even though they're just one cell, they're pretty complex. So life is amazing. And we're using this a lot as we like plan our mission to Mars to see if there's life and throughout our solar system. So getting from the solar system and going to Mars, obviously to do that, we have a method and they use lots of engineering and also the thing called a scientific method. What what I noticed as I was going through most of your papers when we were grading them is, and sometimes we still get confused on this, independent and dependent variables, and how we graph those and how do we decipher what is our independent and dependent variable. Way I kind of explain it to my kids is, especially teenagers, is everybody wants to be independent. Our independent variable doesn't depend on anything else. So that is the thing we're always changing in our experiment. The independent is what we're going to change. The dependent is what we're going to measure. So when you're trying to identify those in a problem or a lab report, just remember independent is what we're changing, and dependent variable is what we're going to measure, because the dependent variable depends on what we change for the independent variable. So does that make sense, guys?
Yep.
Okay. And so as we look at this, another way to think of graphing them too is on the x-axis, our dependent variable is always on the x, on the y-axis, and on the x-axis is our independent variable, what we're changing. And a way I try to think of that is, you guys know how little kids are dependent? They reach up to hold a hand. And so if you remember y to the sky, our y-axis is always going up and down, our x-axis is horizontal. Y to the sky, our dependent variable, always is going to go on our y-axis. So if you think of a little kid reaching up to hold a hand, that would be an example of they're dependent, and so our dependent variable always goes on the y-axis. Does that help a little bit?
Yep.
Okay, so if we looked at this graph and they said we were trying to figure out what would our dependent, the graph, can you guys see it right there, is on tadpoles and pH. Why would our independent variable be the pH? Because we're changing it, right? And the dependent variable is the number of tadpoles. We're measuring the number of tadpoles. So the pH is what we're changing, and the tadpoles are what we're measuring. And so most of the time when you look at a graph, you can even just look at it and determine your independent and dependent variables. All right, as we moved on then, we got to get into cells and the cell theory, and we did a little work with people to discover the cell theory. And so make sure you know a little bit about the guys that discovered it, know about Hooke, Virchow, Schleiden, and Schwann, and make sure we did a little matching activity, I know, throughout the course where we did that. One way to determine the difference, I think kids a lot of times, and this is kind of funny, I'm going to use a company to tell you about this, but Schleiden and Schwann were the first guys to recognize plant and animal cells. So my kids always get confused on which one did which. So Schleiden did the plants and Schwann did the animals. And this is a silly way I remember it. I really like ice cream. And the Schwann man, do you guys know what the Schwann man is? That delivers ice cream in frozen trucks. Have you guys ever seen those? Huh?
Yeah, we order them every Tuesday or every 2 weeks.
Okay, so he delivers ice cream, right? Ice
Yeah.
cream comes from animals most of the time. And so Schwann discovered animal cells, and Schleiden discovered plant cells first. So that's just a little way to maybe help you. I'm not saying that, I know the Schwann man delivers plants too, but if you think of ice cream, the Schwann, so the scientist Schwann, I don't know if they're named after the same thing, but he discovered animal cells. So Schwann discovered animal cells, and Schleiden discovered plant cells. So that might help you just with that one. And then just be familiar with the cell theory, that cells are the basic unit of life. We can't make cells. They just don't show up. They have to come from another cell. And then all organisms are composed of cells. So make sure you know that. And then as we look at it here, make sure you know the difference between a prokaryote and a eukaryote. And here's another little memory thing to help you. I like memory clues to help me. And what they say right here is a prokaryote has no membrane-bound organelles. Now, this is not true, but this is a rhyming thing to help you remember. Pro means no. Prokaryotes have no membrane-bound organelles. Pro doesn't really mean no, but it's a way to help us. So pro, no, it's just a rhyming thing. Prokaryotes have no membrane-bound organelles. They do still have DNA though, and it's in a little circle, but they do not have a nucleus. They have no membrane-bound organelles. Most of the prokaryotes, or all of them, are bacteria. This is another fun name you could call your brothers or sisters. You could be like, you are a eukaryote. And if they don't know that, they could get mad, but really, that's kind of a compliment. We have membrane-bound organelles. And so you are a eukaryote is the way you can remember it, and you do have a nucleus in your cell, and you have membrane-bound organelles. And so those are little memories. Pro means no, which it really doesn't, but pro means no membrane-bound organelles, and you are a eukaryote. So just remember that when you have to identify them. I would be familiar with every one of the organelles and their jobs. And you can make, there's some fun songs I posted in our updates to help you remember those. But I listed some of the basic ones here. I know you guys can read, so I'm not going to read them to you. Really, really important one, though, I would remember, we're discovered that just last unit we worked on the ribosomes make protein. I would know that for sure. And the mitochondria gives us energy. It's the powerhouse where cellular respiration takes place. We did that in the last unit. And chloroplasts have, that's where the magic, where we turn sunlight into sugar in that amazing process called photosynthesis that supplies all energy on life. If we didn't have photosynthesis, none of us would be here. So make sure we understand that photosynthesis is very, very important. And we'll go over those in a couple of slides. We can look at this, know the difference too between plant and animal cells. This is just a quick little difference that you might be asked this on the final. The plant has a cell wall, has chloroplast, and a large central vacuole that stores all its liquid, which makes sense because plants can't go around and drink water or get food. So they store a lot of their stuff. And so they have a large vacuole. Animal cells, we have bones to support us. We don't have cell walls. And we have no chloroplast or no vacuole. So, any questions about that?
Nope.
Good thing. Okay. So, as we look at the rest, we also got into a little bit of chemistry. You cannot study biology without chemistry. So I did put on a slide because I think most of you guys did pretty well with this, but make sure you know atoms are made of protons, neutrons, and electrons, where the protons and neutrons are in the middle, electrons rotating on the outside, and that the number of protons is the atomic number. And then once we went through that, we got into what are we made out of, and like what do we eat? And so you guys did that lab where you looked at all your food and what we ate and kind of broke it down. So the big four main molecules of all life that we know of Earth is carbohydrates, lipids, proteins, and nucleic acids. Some scientists predict that like life on other planets might not be carbon-based, so it might not be these same things. Which sounds kind of like weird and sci-fi to us, but some people think actually that some scientists have done some studies that maybe silicon could be it, or arsenic. There's just these different theories of what is all life actually carbon-based. But our life on Earth, we have carbs, which are our main source of sugar, and they provide glucose. We have lipids, which store our energy. And we have proteins, which make up like all our muscles and structural components, which are made of amino acids. And we learned also later on after in one of the units how DNA goes to RNA to make protein, which gives us our three-dimensional structure and what we look like. And then that jumps into DNA and RNA, which are the instructions, and they are called nucleic acids. So make sure you're familiar with those and can identify those, like what are our food sources. We looked at some of those. What what do we know? What are carbs used for? What are lipids? What are proteins? What are nucleic acids? And what are their building blocks? And I know you guys analyze that with your food and looking at some of the stuff. So that's really important to know. And then we also looked at enzymes. And we did this with our potato lab. And so where you guys experimented with the potatoes at different temperatures, we were looking actually at enzymes. How does the enzyme act at different temperatures? And really what happened in that lab is when the potato was really hot, we killed the enzymes. And so that's why the hydrogen peroxide didn't react. But each enzyme is very specific and it works to speed up a reaction. The saltine, like if you've ever tried to eat a whole bunch of saltine crackers, why do you guys think it doesn't break down? It's because we run out of spit. It gets like this dry wad, right? Like this nasty wad of crackers in our mouth. And that's because the enzymes, if you actually took a cracker and just put it in your mouth, your saliva has enzymes that start breaking that down right away. Well, when we overload it and the enzymes can't get to all the cracker, we can't break it down. And so enzymes speed up stuff. That's how we eat like suckers and candy that we suck on. It's the enzymes break it down in our mouth before it even gets to our stomach. So enzymes speed up chemical reactions. So a way to remember that is remember, like if you put too many crackers in, the enzymes can't get to the cracker and it doesn't break down. And so just remember, but they're very specific to what they break down. And we have tons of different enzymes in our body that are going to react with different things. But temperature will damage them and extreme pHs. So moving on to the next one. This is one of my favorite units, but we have these amazing cells and we have to make more of them. If we don't make more, we're not going to live. Number one, we have to grow. We have to grow from an infant to get bigger, but we also have to heal. Anytime you guys have had a cut or a scrape, you have to heal that. And we do that with cell division. And then the next reason is type of cell division is going to make sperm and egg cells through meiosis to make more humans or more animals or maybe another maple tree that's going to pollinate. We can use it with plants, animals, but we have to have meiosis too to get genetic variation and more species. So, in fact, things to think about in cell division is know the difference between haploid and diploid. I always think about haploid means half as many cells, and diploid means two. So we have two copies of it. Okay. So haploid is half as many, and diploid means twice as many, or two copies of each one. Does that make sense?
Yeah.
Okay. And then if we look, mitosis is the process of how we take a diploid cell and make another diploid cell. It's cloning. It's making an exact same copy. And the cell's going to do that through the stages of, I call it PMAT to remember it. So that might be a little thing if you guys want to write it down to help remember the stages in order. If you think of PMAT, M-A-T, it's prophase, metaphase, anaphase, telophase. Those are the stages of mitosis, which is not the only stage of the cell cycle, but it is where the nucleus divides. So mitosis is the division of the nucleus to make another diploid cell. The other stages are cytokinesis, which is the division of the cytoplasm. And so then after the cell is divided and divided its cytoplasm, it's going to grow again. It's this baby cell, it's going to grow. Then it's going to go to the S stage, which is going to double its DNA. And it's going to go back to the G2 stage again, where it's going to grow and then go through mitosis again. So it's always constantly going through the cell cycle. And they say our skin cells do this so fast that they never, they just keep doing it. And they say even after we die, our skin cells are still dividing for a little while, like 24 hours after we die, because we're constantly shedding and replacing skin cells. Any questions about mitosis? Did I do that too fast?
No, that's good. That's good.
So meiosis is this totally amazing process that I don't know. It's what makes you you, and it makes us all unique. The chances of anybody, unless you're an identical twin, having the exact same DNA is very slim because of this process of crossing over an independent assortment. So if you guys can see that image right there, there's like these two chromosomes, these with their 4 chromatids right there. And they do this amazing process. And this is inside each one of us. And we get 1 copy from our mom, 1 from our dad. Well, then our mom and dad's DNA combine and switch around. And then they give each other's part of that DNA. And then they divide into 4 cells. And each one of those cells could either be a sperm or egg cell. In females, every time they go through oogenesis, which is make an egg cell, only 1 cell becomes an egg cell, which is kind of unique, and the others kind of fade away. But that's in humans. And this process lets this whole unique set of chromosomes make you. And then when a sperm and egg come together, it's a combination of your parents' ancestors that have been recombined, and it's a pretty cool process that makes us so unique. And this genetic variation is huge for survival of species. Like, it gives everything a better chance of survival because we're not all the same. If a disease came by to wipe people out or wipe something out, some might be resistant. A good example of this in Idaho is blue tongue with the white-tailed deer population. Every few years, it wipes it out. Have you ever heard of that, kid? Blue tongue with white-tailed deer?
More and more. Yeah, kind of.
It wipes out a lot of the population, but some will survive because they're resistant. And a blue tongue is just a little disease spread by fleas, and it kills white-tailed deer. But some deer are resistant, so we keep getting white-tailed deer. If we didn't have genetic variation because of meiosis, it would wipe out that species. Does that make sense?
Yeah.
Okay. And so this goes through. What I really want you guys to get away from that is I probably went off too much on meiosis, but meiosis will make 4 haploid cells. Mitosis will make 2 diploid cells. And meiosis does this by going through the process twice. So make sure you understand that. If you have any questions on it, let me know. Does that make sense, kid?
Yeah.
Okay. I know this is a hard question, and the next part of one of your units was, how does stuff move in and out of a cell? Like, it's a cell. How does stuff move in and out? And so to think of how does this work, we did cellular transport, and we did the difference between passive and active transport. And remember, the cell membrane is like the security guard. It only lets certain things in and out. Not everything can pass through the cell membrane because it's selectively permeable. And we have the selectively permeable membrane that can transport stuff in different ways. All of these types of transport on this slide do not require energy under passive diffusion. So those 3 numbered ones right there, diffusion, osmosis, and facilitated, require no energy. That is passive. It means kind of like just chilling back. And a way to remember this, it's going to move stuff from a high to a low. So if you think of like riding your bike downhill, you're going from a high surface to a low surface, and it doesn't take very much energy. So all of our passive transport is moving from a high concentration to a low. It's just what we're moving is different for the 3 different things. So for diffusion, it's small molecules that can just go through the cell membrane. Osmosis is just water. I know quite a few kids miss that on the test. What is the difference between diffusion and osmosis? Osmosis is just the movement of water. Diffusion is any particles that are very small that it can go through. Facilitated diffusion is for like big things that need a protein channel or a channel to get through. Sometimes things are too big, and so our cells have actually made little channels in the membrane that will let certain things go through it. And so it's still going from a high concentration to a low, but it has to go through a channel. It just can't go through the membrane because it's too big. Then the opposite happens with active transport. Active transport is very active. It's like riding your bike uphill. We're going to go from a low concentration to a high concentration, which is going to take energy. We know it takes a lot more energy to go from a low to a high, and that just helps you remember that we go from a low concentration to a high with active transport. So I hope those little hints in the bicycle, if you think of riding your bike, will help you with passive and active transport. The next unit is, will you answer it, please? Go answer it, Riley, and tell me. Sorry, Caden, it's kids too.
You're good.
If it's an emergency, I'll have to go answer it. But as we're looking at this right here, we have photosynthesis and respiration. So I kind of went over this a little bit before. So photosynthesis is a process that captures sunlight. And so make sure we know it. Is there somebody there? Oh, you can bring him in. Just tell him I'll be just a second, like 5 minutes, 10, 15 minutes. Sorry. Sorry.
Yeah.
Photosynthesis is a process that is going to allow the plants to capture energy in chloroplast. Make sure you guys know the difference between the two reactions. There's light reaction and dark reaction. The light reaction is needs sunlight to fuel it and get going. And the dark reaction is actually going to happen inside the chloroplast. And it doesn't need light, but it's going to produce a lot of our energy. Does that make sense?
Yeah.
Okay. Few more slides here. Are you doing okay still?
Yep. I'm just filling this out.
All right. So the next slide I have for you guys is cellular respiration. This is our amazing process how we take that stuff that got captured by the sunlight and we make sugar. And it's just a super cool process that's going to use energy. And there's two ways we can do this, cellular respiration. Some organisms don't use oxygen, which means anaerobic. And some organisms use oxygen, which is aerobic. And so aerobic, I like to think of the exercise aerobics like we use, right? It requires oxygen. Make sure you look at this table, guys, and know that aerobic will produce 38 ATP, whereas anaerobic doesn't produce very many. And so that's important to know. The steps, there's 3 steps to the cellular respiration. And I will tell them to you guys right now, they're kind of on the slideshow right here, but it's glycolysis, and then it's the Krebs cycle, and then an electron transport chain. So that's an aerobic respiration. In anaerobic, it's just glycolysis, and then it does fermentation. So does that make sense, Kaden, on the respiration? Main things I would remember about that is ATP is the form of energy, and that cellular respiration, aerobic produces a lot more energy than anaerobic. And our next slide was on the ecology unit. I loved watching your guys' projects that you guys submitted. I thought those were really interesting with the PowerPoints and stuff. That was really fun. Please make sure you know the levels that things form, that organisms are the very basic, and they make a population that's all the same organisms. And then a bunch of different organisms make a community. And then a community makes an ecosystem. And then ecosystems make our living biome, our area. And then the biosphere is all living things on Earth. And so we study all this with ecology, which is the interaction of living things. And we did that rabbit lab that looked at how do things live and what different things make it work. And so we have some different things, and just make sure you familiarize yourself. Food webs. Make sure you guys all know that food webs start with the sun. Everything, if we didn't have sun, we would have no energy. And then it goes from producers to the primary consumer, to the secondary consumer, to a tertiary. So if you did that food web and you didn't put the sun and you didn't put 10 organisms in, redo it so I can give you full points for it, because I want to give everybody full points for everything. I want you all to have the highest grade possible before you go on your final. Does that make sense?
Yep.
Okay. All right. So next unit, this one is a little complex, but process of taking DNA, RNA, and making protein. It is just an amazing, amazing process. Some things, look at the comparison. We did an assignment on comparing DNA and RNA. RNA is one strand, DNA is two. Make sure you know that. DNA is replicated inside the nucleus. DNA never leaves the nucleus. It is the boss, and it does not leave the nucleus. It's kind of like a boss in its office. It never leaves the office. And then RNA is the messenger. It comes out, and it gets all the work done. It's like the worker bee. And so we have three types of RNA. We have M, T, and R, which stand for messenger, transfer, and ribosomal. These are made by reading DNA through a process called transcription. And then these three are going to come together and make a protein through translation. And so this is a pretty cool process that's going to make us. It makes our protein. And to me, how we know this is amazing. If you guys can look at that little sheet right there, it shows you the process. We have DNA. We do transcription. We make mRNA. It takes the message out. And it comes together with the ribosome and the tRNA. And then it makes protein. So pretty cool. Make sure you know RNA has U, no T, but has uracil and no thymine. That's really important. Any questions there?
Nope.
Okay.
Pretty good.
Pretty good? Okay. Awesome. The last thing is genetics. Oops, I skipped two slides here. Genetics is really clinical process. I want you to make sure you guys know how to fill in a Punnett square. So some of you guys, I know it's hard on that Word document, and I'll get that fixed. But T, make sure you just bring them together, these two boxes. So I did big T, little t, big T, little t. And then also, so you'll just fill that in. And then all of these plants would be tall because the dominant gene is there. If the dominant gene is there, it is going to be tall. Okay? And so if it was small, two small letters, like this one right here, then it's going to be recessive. And so that's where it would be a short plant. But if there's big T, little p, T, big T, little t, it's going to be tall. What this is this is the genotype. This is what are the genes of that plant. Genotype is a genetic makeup. Phenotype is what does it look like. And I would think it's a picture. P for picture is what does it look like. I can't tell what anybody's genes are, but I can tell what they're expressed. And so if you were going to look at me, you could say I have blue eyes. So that would be a phenotype. You can't tell me my genotype because you don't know what I have. Most of the time, blue eyes are recessive, so you would know I had little b, little b. But we don't always know people's genotypes, but we can see the phenotypes. And last thing I want to go over, and I'm getting some confusion on this, is a little more complex genes. And the big one is XY. XY means boy, XX is a girl. And certain characteristics are carried on these chromosomes. So an X chromosome might carry it, and if the X is only present, it's going to be expressed. So if you look right here, this is a boy where I'm circling. Can you see where I'm circling, Caden?
No, you can't really see it.
Let me try it. So if you look at this right here, now, well, that's a boy because it's XY. So this is the Y chromosome right here. This is a boy, right?
Yeah.
This boy is going to have the disease right here. Do you see the one I circled? Really poorly, yep. But I did. That's going to have the disease because it doesn't have a Y to cross it, cancel it out. So this male would be colorblind. This female over here has the disease on, well, let me look real quick. And the mother must be a carrier. So on this one, if we looked at it, this one, the female has genes to block it out, and so they would not have it. Does that make sense? And I actually circled the wrong one. The one that would have the disease is the bottom one. Let me change the color of the pencil. And so this one would have the disease. And so does that make sense, Caden?
Yeah.
Okay. Do you have any other questions for me, Caden?
No, I'm probably just going to fill this study guide out, and if I have any questions, I'll just let you know. So far, it's pretty good.
And I will give you bonus points for attending, and I think there's a few others here. It sounds like Brian, Chloe, can you guys hear me? You can just type too. I will give you guys bonus points for attending, and anything you guys need help with, please reach out to me. Get your grade up, get it to 20%, and because the final's worth 20%, get it way above 20%. But I want you to like think of that. So when we do our final, you have the highest score possible. And please just keep submitting assignments. Okay. Any other questions you guys have? You can type them in the chat if you do. So if nobody has questions, just give me a thumbs up in the chat that you're good to go, and we'll be done. Okay. And call me, text me, or email me if you need help.
All right.
All right. Thank you guys for attending. I'll give you bonus points for attending.
Thank you.
All right. Thank you.
Thank you.